lecture series as a part of Time Teach for Science program, which is being organized in collaboration with INYAS and Prayas. So again, I'll give small details which I have been repeating for the past 3 lecture series. This Teach for Science program is an initiative taken by Prayas in collaboration with INYAS, where students and teachers from across India are mentoring other students and teachers on various projects, and we have about 30 to 40 mentors and mentees in this program. As a part of this program, students are expected to attend these lecture series, which we organize every Saturday at 11 a.m. And this is the 4th one in this series. I think 1 or 2 might be coming up before the program ends. Today we have a very special speaker, Dr. Rifat John. She is from Plant Molecular Biology Lab at Department of Botany, University of Kashmir, Srinagar. So welcome, Dr. John.
Thank you.
I'll briefly present her biodata before she gives us a very interesting talk on how do plants respond to environment. She has a PhD degree from IIT Delhi and a master's degree from Jamia Hamdard University. She, before joining as an assistant professor at University of Kashmir, she was assistant professor at Government Degree College for Boys, Anantnag. And she has won a number of fellowships and awards, including Indo-US Genome Engineering and Editing Technology Overseas Fellowship in year 2017. She was selected for INSA International Bilateral Scientist Exchange Program to visit Hungary in 2016. She was also member of INYAS from 2016 to 2021. She was also awarded Young Scientist Award by Jammu and Kashmir State Science Technology Council in 2015. The list is really very long. I'm really overwhelmed that she is part of this speaker for these lecture series, and she has completed a number of research grants, including 3 which have been completed and 2 are ongoing, and she's actively involved in activities outside her research and teaching domain. She organizes National Science Days and is also member of Department of Research Committee at University of Kashmir, member of Anti-Ragging and Discipline Committee for Women Scientists, and so on. So it's a privilege, Dr. John, to have you here, and I'm looking forward to such an interesting talk. Kindly, so it's all up to you now.
Thanks, Bindya, for your nice introduction. Okay, I'll start my today's talk. How do plants respond to environment? Since I work in this area, my main research theme is to understand how plants respond to different abiotic conditions, stress related conditions. So today I'll be talking briefly about this. We know that, am I audible to everyone?
Yes, perfectly fine.
Thank you. So we know that environment on the globe, it's very different. At some places we find marshy areas. We have deserts wherein there is high temperature and water scarcity. We have saline soil, high salt concentration, for example, near the coastal areas. And similarly, we have conditions like high altitude mountains, snowing there. This is the picture from the Kashmir. This is the deodar plant surviving in extremely cold condition, -10, -18 degrees. So we find, there are varied environmental conditions on the planet, and still plants grow everywhere. In one way or the other way, we find plants growing. So it's very important for us to understand how plants tolerate this extreme conditions, environmental conditions. Now, if we categorize the response of the plant to the environment, we broadly classify response into 3 main categories: avoidance, tolerance, and escape. Now, what is avoidance? What do we mean by avoidance, and what do we mean by tolerance, and what do we mean by escape? First of all, escape. Escape means plant completes its life cycle before the onset of the unfavorable conditions. Means plant does not face unfavorable condition at all. Means plant grows flowers in the in March, April, May, and before there is autumn, plant completes its life cycle. It's a very short life cycle. Epihedral plants, 4, 5, 6 months they survive, and then they complete their life cycle. They don't face any environmental harshness. Means escape. Now there are some plants which try to avoid. Now, what is avoidance and what is the difference between avoidance and tolerance? Avoidance means plants adapts itself, conducts itself in such a way that it does not let unfavorable condition to stun its growth, to harm it. For example, you will ask how. Now, let us imagine there are, at high altitude mountains or in an open area, there are a lot of radiations, high heat. Now, plant will orient its leaves or its canopy in such a way that it will not face all those radiations. That is avoidance. Plant will store water so that it does not face that drought-like condition. That is avoidance. Tolerance is another mechanism wherein a plant tries to modify itself to overcome unfavorable condition. So plants survive in an unfavorable environmental condition by both mechanisms, avoidance and tolerance. So we know that there are a lot of stresses in the environment. There are water stress, which is one of the most important abiotic stress that affects the plants, and especially in this current world scenario wherein there are water wars uh, on water, related to water disputes. So this is one of the important, in coming years also plants are going to face, and global changing global climate, all that has led to the that drought conditions in various parts of the world. So this is one of the important abiotic stresses which affects the plant growth. We have temperature stress. For example, if a plant has too cold or too hot, both the conditions are not good for the plant, because hot weather can affect plants adversely. Intense heat can cause plant proteins to break down, denature, and there are other stresses. For example, heavy metal, and heavy metal pollution, as well as there are air pollution also. All that affects the plant growth, because you might have seen in and around Delhi, thick layer of that soot on the leaves of the plants, that leads to the decline in the agricultural yield, that in fact affects the plant growth. And similarly, there can be radiation stress. For example, too much of photosynthetically active radiations or too much of UV radiation can affect the plant growth. And on the other hand, we have biotic stresses as well. Now, biotic stress is the kind of unfavorable condition wherein living organisms such as fungi, bacteria, insects, weeds, weeds also are a part of biotic stress, because weeds, why weeds are harmful to us, or why weeds are unwanted, because they compete for the nutrition, they compete for the space with the plants which we grow for our purpose, for food purpose, for other purposes. So weeds are considered as unwanted and unprofitable plants, that they inhibit the growth of the desirable plants such as crops or flowers. So broadly, if we can classify response of the plant to different environmental conditions, is escape, wherein there is plant completes its life cycle before the onset of the unfavorable conditions. There we have avoidance, where plant restricts the impact of the unfavorable conditions, as I said, changing the orientation of leaves, having some plants have very deep, they develop the deep root mechanism, deep roots, so as to absorb as much as water they can. Tolerance is the plant's ability to grow and produce an acceptable yield despite there are harsh environmental conditions. So avoidance and tolerance are the two mechanisms by which plants grow in an unfavorable condition. In fact, if we look at the history of evolution, plants have developed wide variety of, highly sophisticated and complicated mechanisms to sense this stress, to sense that environmental cue, to respond to it, and then to adapt. So different responses occur at different levels. For example, plants respond at molecular level. By molecular level, we mean changing the proteome content, changing the different proteins, stress proteins, synthesizing different proteins. We have heat shock proteins, for example, or changing the tissue concentration at anatomy level when we dissect the plant growing in normal condition, growing in harsh condition. And when we do the anatomy, we do the tissue section, we can clearly see that there are changes in the anatomy. For example, it will develop sclerenchymatous tissues. It will suddenly develop the, chloroplasts as a modification to the environmental change, or they can adjust the membrane system. For example, in response to cold, we see that there is change in the fatty acid composition of the cell membrane, because in response in this cold temperature, these membranes, they develop more and more unsaturated fatty acids to keep it in the fluid state. Similarly, there are changes in the cell wall architecture, change in the phenyl propanide pathway, change in the growth pattern by altering the cell cycle or rate of cell division, and there are changes in the metabolic pathway as well. So plants respond at different levels, at many levels, to adjust itself, to tolerate, to avoid the stress, and grow, and grow to acceptable yield. One of the mechanisms, physiological mechanisms, is osmoregulation. All of you must have heard about osmosis, turgor pressure, which are very important for cell survival, cell growth impact. Am I audible?
Yes,
Am I audible?
Yes.
Thank you. Because so osmosis, I'm sure you all must be knowing. Now, one of the challenges for the plant under the stress condition is to maintain a proper osmotic balance, and plant strikes the balance by osmoregulation. Now, what is osmoregulation? How do we define osmoregulation? Osmoregulation is an important process in both plants and animals as well, as it allows organism to maintain a balance between the water and the minerals at the cellular level, despite there are changes in the external environment. Now, if we talk about the plants, I will briefly tell you here how plants perform osmoregulation. When there are, for example, you imagine a plant growing in a saline condition. Now imagine a plant, you're not watering a plant. Okay, you stop watering the plant, and plant is having a drought-like condition. What will plant do? Now, what will plant do? It will synthesize compatible solutes, which we call as osmoprotectants. Osmoprotectants are compatible solutes which are osmotically active, which help in maintaining the osmosis inside the cell, but at the same time they do not affect the enzymatic activity or normal metabolism. They do not affect, but they only help the plant cell to maintain a normal osmotic balance. So what is an osmolyte? It is one of the important metabolic consequences of osmotic stress. There is accumulation of osmolyte. These are low molecular weight organic compounds, and they are highly soluble. They do not interfere with the normal metabolic reactions because they are non-toxic, even they are at high concentration. So we see in the lab, if you do an experiment, your MSc student or your BSc student, if you do an experiment, you take two plant sets. One, you water, and another plant you don't water, you give it a drought condition. Then you take the leaf tissues and you check the concentration of, let us say, proline or carbohydrates or antioxidant enzymes. You will see that the antioxidant enzymes are for the reactive oxygen species quenching. Okay, if you see that levels of amino acids or polyamines or glycine, betaine, proline, you will find that the plant set which has been given stress will have higher levels of proline, will have higher levels of polyamines, will have higher levels of glycine, betaine, GABA as compared to the control plant which is going normally. Why? Because plant synthesizes these osmolytes in response to the stress, because because due to the stress there is change in the osmotic balance. Plant has an osmotic shock because it has less water, so there will be there is tendency of exosmosis. Plant may lose water, cells may lose water because there is drying condition. In that case, what does plant do? It is a strategy. It will develop these osmolytes and will not lose the water and will try to avoid that condition. And let us say another condition. Let us take, and I'm briefly giving you examples. Light stress. Now, light, what happens, if there is too much of light, photosynthetically active light, see, it can photo damage, right? It can damage the photosynthetic pigments or thylakoid structure. Now, what plant has? Plant has a xanthophyll cycle. In the xanthophyll cycle, what happens that chlorophyll excited, if you can see here, this chlorophyll gets excited due to the light energy. Now, this is excited chlorophyll. It will pass on some energy to zeaxanthin. Now, this zeaxanthin will get converted into anthraxanthin, and anthraxanthin will convert into the violaxanthin. In this excess amount of light energy which is absorbed is radiated as heat or as fluorescence. When there is no light or there is no stress, violaxanthin will convert back into the anthraxanthin, and anthraxanthin will convert back into the zeaxanthin. So plant, this is another mechanism by which plant tolerates or avoids the light stress. So we saw osmoregulation, we saw this photo inhibition. Another, you can say, we can imagine, or we can talk about the stomatal closure. When there is unfavorable conditions, such as when there is high heat or there is low water, what plant will do is it will close its stomata. Automatically it will close its stomata so that it does not lose its water, and so that there is no more transpiration, and plants tries to conserve the water. So stomatal control of water losses has been identified as an early event in the plant response to the water deficit and field conditions, leading to a limitation of carbon uptake by the leaves. आप ये experiments जो है lab में कर सकते हो, your UG PG students. These experiments we conduct for our PG students, for our UG students. You can do these experiments. You give one set of the plant, keep it at controlled normal condition, another plant, give it drought or give it high light or high heat, then you try to take the leaf and see the stomata in its leaves. You will find that stomata are closed. So I'm sure you must be knowing how stomata open and close. There is a molecular mechanism behind it, uptake of when it is more turgid, it will open. When they are flaccid, they will close. And let us say another example of how plants modify themselves or respond to environment is phytogelatins or phytokeratins, we can call it, howsoever you pronounce it. Now, phytokeratins, what are they? Phytokeratins, they chelate ions or heavy metals. Plants growing in heavy metal conditions, they chelate with these. They synthesize proteins. Okay, and now these proteins wrap themselves around the ion and they bind to form a complex chelating molecule. And then what happens that this ion, it does not now pose any threat inside the cell, because iron, this heavy metal, it has a varied oxidation state. It can react with proteins, it can react with other ions. Now these proteins wrap around this ion and then make it less toxic, and then it is translocated to the other parts. You must have heard this in phytoremediation, when plants are used to take up the heavy metal and make the site clean. This is one of the strategies for bioremediation. So now the question is, important question in front of us is, why we have to study the stress? Why we are interested? I mean, why should I study stress in the plants and how plants respond? I mean, why should I bother myself, or why should we put in a lot of energy into this? Now, we know, for example, this is the recent data, which I, just before this presentation, I got from the internet, that there are, global hunger index, all across the world, all the countries are ranked. And if we look from our country's perspective, we come under, we are seriously, this GI determines hunger index, and India was classified in the serious category. Serious category means we have a lot of people, a significant number of people, you know, facing hunger. Now, let me tell you, hunger is not about only having, full meals. No, there is another hunger, which means even if we are taking food, but it does not meet our nutritional needs, that also comes under the hunger, right? So, because of this global hunger, hunger is at the global level, and especially India is also facing this issue of people, going to bed without food. At the same time, population is increasing and land is shrinking because of the overpopulation. Because of the increasing population, we have to make more roads, we have to make more houses, and as a result, the land which can be used for agricultural purpose is now used for urbanization. So we have on one hand, this is an unfavorable condition. On one hand, we have increasing mouths to feed. On the other hand, we have land shrinking. So this is a double-edged sword kind of a thing. So the responsibility of the agricultural scientists is to see that how they can produce more in the less. So what is idea is that all these areas which we cannot use for agricultural purposes, to make crops so that they grow in these conditions, they grow in the harsh conditions, so that we can have more agricultural produce. And one of the promising approaches to improve the ability of the plant to cope up with the environmental stress is to combine the utilization of vast biodiversity of the plants we have and their wild relatives, and try to understand to use them in the modern breeding programs, use these elite genotypes which are growing in extreme environmental conditions, to use those genotypes for breeding programs. Or we can isolate proteins or genes which are important to give the plant stress resilience, and we can take that gene and put into the vulnerable plants. So global programs such as Global Partnership Initiative for Plant Breeding Capacity Building aims to select and distribute seed crops and cultivars with tolerance to abiotic stresses in order to facilitate use of plant genetic resources for food and agriculture. I will briefly, I will talk about my work, what we do at Department of Botany, University of Kashmir, what my group is involved in, how we are trying to, because, the program officer who wrote me into this program, she told me, I asked her, Pooja, what should I speak? What I'm going to speak? She said, Rifat, talk about your work. So I have briefly introduced you to the topic, and now I will be talking about what we are doing at, our lab. Now, for example, this is one of the work of my PhD students, molecular characterization of Zea maize from Kashmir Himalayas for low temperature prawns. So what we did. Now, all of you know that, you must have heard about the maize growing in, very hot climate, I'm sure. Right. Now we have we have a maize which is growing in Gurez. I don't know how many of people you have heard about the Gurez Valley. It is a mountain. It's a Himalayan mountain where 2,580 meters above sea level, right? And this corn, these tribals grow there. If you can see this, am I audible? Guys, are you listening?
Yes.
Okay, thank you. So, for example, you can see in this picture, these are the tribal people and they are growing the corn. Now, this corn grows, at very cold temperature, chilling, snowy water. This corn grows. And what we did is we took this maize and compared it with the maize growing in Gujarat. Now, these are two different, obviously two different geographical locations with different climatic conditions. Gurez, very cold, right? If you have visited the Kashmir and if you want to go to Gurez, you cannot go after August because all the roads are closed by snow. The road is open only for a couple of months when there is high temperature, and because the snow melts and roads are clear. Otherwise we cannot go in September or October. Road is closed almost. So we took this maize from Gurez Valley, and we took maize from Gujarat, and we tried to compare how this Gurez local is able to grow in cold climate. Okay, now maize is a subtropical cereal crop, and it is cornerstone of global food supply. I'm sure you must be knowing this. India, fifth in term of area and third in term of productivity in case of maize. So why we did this? For the extension of vegetative period of life cycle of maize, and we try to understand how come low temperature tolerance is present in this Gurez local. What makes Gujarat maize GM6? What makes Gujarat maize susceptible to low temperature? Why it cannot grow at low temperature? Because we saw that this Gujarat maize in our growth chamber, it was not able to grow below 20 degrees. Once we gave it 20 degrees, it showed signs of senescence. So first of all, I'm sure you must be knowing that in maize, this low temperature tolerance, we know that it's a complex trait. हम ये नहीं सोच सकते हैं कि that low temperature tolerance is because of couple of 2 or 3 proteins. No, it is a complex trait. So it employs physiological molecular approaches. We try to understand how does plant tolerates this low temperature stress. So plant has a Kranz anatomy. I'm sure you must be knowing this. So in Gurez local, what we found is even if it is tolerant to low temperature, but we saw that it has low yield. ये बहुत ही कम seeds produce करता है. So temperature and precipitation are the two important climatic variables, where radiation being under critical force that affects the yield. And in Gurez local, we found that it has low yield as compared to Gujarat maize. No, so our hypothesis was that adaptive low temperature tolerance in Gurez may be attributed to antioxidant defense system. So what we did is, first of all, we evaluated maize and compared it with Gujarat maize 6. So on one hand, we have Gurez local, which is low temperature tolerant. We have GM6, Gujarat maize 6, which is low temperature susceptible. And then we compared the two. So how we took the study step by step. First of all, we did a physiological study. We try to see the levels of proline, presence of sugars, increase ho raha, nahi ho raha, glycine, betaine. We try to understand, how much antioxidant enzymes are increasing. And then IRGA, we did infrared gas analyzer, is an equipment by which we don't need to cut the plant. Plant is growing in the pot, and we see photosynthetic efficiency, carbon dioxide assimilation, and other parameters. So this is our growth chamber wherein we give the plants maize. We got them from the SCOST, Agricultural University for Science and Technology, and we grew them in our department. These are Gurez local, and this is the Gujarat maize 6. We gave it different temperature. For example, control we kept at 25 degrees, and then we gave the low temperature stress of 6 degrees, right? Similarly, after 4 hours, after 6 hours, 8 hours, we took after 2 hours, we took the sample. Similarly, Gujarat maize, we kept at the control. Then we also gave it 6 degrees and took the sample after 2 hours, 6 hours, 4 hours, 6 hours, 8 hours, 10 hours, 12 hours. So we took every 2 hours the samples, and we assess them for their physiology, for their molecular response. Number 1, so we try to evaluate differentially temperature tolerant maize genotypes for various biochemical and antioxidant markers. So we took the maize seedlings, okay, up to 3 leaf stage. All right. Then temperature, a short-term stress was given of 6 degrees. We considered the cold stress for the plant. देखिए cold stress जो है, it varies from plant to plant. We gave 6 degrees, right? It is for the maize. Maybe for some plants, 6 degrees is not a cold stress for that plant. Maybe 10 degrees is a cold stress. Or maybe some plants, 1 degree is a cold stress. So this temperature varies from plant to plant, right? We cannot generalize that 6 degrees is the cold stress for every plant. It varies from plant to plant. So we did the literature review and try to understand which is the cold stress for the plant, and then we zeroed in on the 6 degrees centigrade as the low temperature exposure for the maize. And we evaluated different parameters. We estimated hydrogen peroxide production. Okay, and we did the histochemical detection of this hydrogen peroxide concentration by studying those leaves. We tried to understand the concentration of MDA, malondialdehyde, which shows the lipid, membrane degeneration. And we assess the activities of various reactive oxygen species, scavenging enzymes. We estimated various osmoprotectants. Just I spoke about the osmoprotectants. We checked the concentration of glycine, betaine. We estimated phenolic content. Phenolics are also antioxidant activity. They have osmoprotectant activity as well. We assess the protein levels, proline, sugars, trehalose also. And then we check the amount of chlorophyll A, chlorophyll B, carotenoids, and anthocyanins. So this was one part of the study. Okay, so I will skip this antioxidant pathway. So this was the primers which were designed. We used primer 3 software, isolated the RNA, and then we did the RT-PCR. These are the different antioxidant enzymes. APX stands for ascorbate peroxidase. MDHAR, DHAR, GR, so SOD, CAT. These are different, antioxidant enzymes, and we checked their transcription. We checked whether their transcription varies in these two plants in response to cold stress. GM6, Gurey's local, grown at 25, grown at 25, grown at 6 degrees, grown at 6 degrees. And then we took the sample every after 2 hours. Then we did real-time PCR to check what happens to the antioxidant enzymes in Gurey's local at 25 and 6 degrees, in GM6 at 25 degrees. So we try to understand what happens in both the plants at 25 degrees and at 6 degrees, how both the plants respond to this 6 degrees low temperature stress. And we saw that, yes, there are changes in the hydrogen peroxide levels, there are malondialdehyde concentration. This is the DAB picture. Now you can see you can see at the Gurey's and GM6. You can very well understand GM6 जो है, Gurey's maize जो है, it shows more generation of reactive oxygen species. There is all brown, but in Gurey's there are green pigments intact, which means okay, this is resistant. There is not much damage, or we can say there is lesser damage as compared to Gujarat maize at low temperature. This is the checking of proline we saw in Gurey's maize. Gurey's local, there is higher concentration of proline, higher concentration of total protein, higher protein levels, higher total protein, soluble sugars, and trehalose, glycine, betaine, phenolics. These are different enzyme active, and this is trehalose and trehalose 6-phosphate. We saw that in fact this Gurey's local, I will show in the in the conclusion. We saw that lesser formation of stress markers, increased accumulation of various cryoprotectants, and efficient antioxidant system is an adapted trait in due course of time, which has helped this Gurey's local to withstand low temperature. As I said, I mentioned that they grow in the snowy water, chilling water. You cannot put your feet for more than 5 or 10 minutes in the snowy water. So how these Gurey's local has adapted? It has adapted because of developing or evolution. Across the millions of years ago, this evolution has occurred, and these tribals or local people have, managed to preserve these land races. Okay, this flint type of maize, sorry. So this flint type of maize provides a promising resource. Now we'll ask the question, how will it help us? It will help us because this provides us an important genetic source. We can use Gurey's local in the modern maize breeding programs, we can cross it. We can cross it with the plant which has low temperature, which doesn't have a low temperature tolerance. Okay, we cross the two plants. It will take us time, but in the breeding program, eventually we can develop a line which is cold tolerant. So we got this work published in Physiology and Molecular Biology of Plants recently. Then we try to understand how photosynthesis of these two plants varies at low temperature. So we assessed different gas exchange characteristics in selected maize genotype for exhibiting this differential temperature response. One एक cold tolerant है, दूसरा नहीं है. क्यों नहीं है? Then we try to understand the photosynthesis. So we took infrared gas analyzer, which is present in our department, which has been, I think, generously given to us by Department of Science and Technology or University Grants Commission. We acknowledge their financial support. Our department is, supported by SAP. Our department is supported by FIST. Our department is supported by Department of Science and Technology, Department of Biotechnology, University Grants Commission. We have a good facilities by these funding agencies, and I duly acknowledge their help and financial support. So this equipment costs about 25 lakhs, this IRGA, infrared gas analyzer, and this we used for our experiments, wherein we did different experiments. For example, we did photosynthetic rate. How does photosynthetic rate varies between the two plants? Now you can see in Gurez local, there is low photosynthetic rate. As I said, ये जो plant है इसमें yield बहुत कम है. There is a lot of this plant is not that much high yielding, but it is tolerant. Okay, stomatal conductance, you can see Gurez local has less stomatal conductance. It has low transpiration rate compared to the Gujarat maize. It has less water use efficiency, right? It has lesser FV by FM ratio and other parameters, excitation, energy capture efficiency. The different parameters we studied by using this infrared gas analyzer. Okay, and this is the heat map, scatter plot, statistical analysis my student did. Okay, and our study provides, okay, our study provides a systemic and instantaneous gas exchange profiles of both these maize genotypes, Gurez local and Gujarat maize, through IRGA. Two genotypes. Now, what was our conclusion? Our conclusion was that two genotypes differ significantly in their photosynthetic performance and they exhibit different photosynthetic rates. दोनों में vary हो रहा था. It was found that maize requires an optimum light intensity of 1600 micromoles per meter square per second, and also temperate origin maize it showed maximum only at 20 degrees. Okay? Only at 20 degrees Gurez local grew, but for Gujarat maize we saw that it required its maximum photosynthetic efficiency was achieved at 35 degrees. Okay, now these two genotypes were very much different. Based on this photosynthetic and model parameters, screening for stress tolerance in other crop plants can be carried out. That was a significant, and that is we proposed. We got this published in photosynthesis research. So next, our aim was, now let us go towards the molecular analysis. Okay, now we studied morphology, we studied physiology. Now we try to understand what happens in the protein levels, how proteomic content is changed, what happens to the protein levels. So what we did is, in our lab, we have an established protocol for two-dimensional gel electrophoresis. I have learned this two-dimensional gel electrophoresis. I want to acknowledge my mentor, Professor Niranjan Chakravarti at NIPGR, National Institute of Plant Genomic Research. He has been very generous during my INSA. I acknowledge fellowship by INSA visiting scientist. I was a faculty at Department of Botany, and then I got this fellowship. I got selected for this INSA visiting fellowship program. I went to Professor Niranjan Chakravarti's lab. He allowed me to work in his lab. I standardized this protocol and this 2D proteomics. Then thankfully I got the project from SERB, Science and Education Research Board. They provided me the financial support for establishing proteomic facility at Department of Botany. And this is these are the pictures from our lab. So we did 2D proteomics to understand what critical proteins change during this stress. So briefly, this diagram shows about is a flow chart showing how 2D proteomics is done. First of all, we take the plant material, we isolate the protein, and then we rehydrate with this. We have strips, gel strips with pH range. Okay, and we had here we have 4 to 10 pH range. These are the gel strips. We put protein on the protein. We put these gel strips overnight. Is passive rehydration. Then next day we come take this strip and put in the isoelectric focusing. Right now, this is one dimensional, because here in proteins will get electrofocused in response to the electric current. We have to standardize the program, and the next day we run this over the gel. So that is why it is called 2D, two-dimensional. One-dimensional because according to the charge, number 2 according to the molecular weight, right? So this is 2D. And then we have the software wherein we analyzed these spots, and then we excise these spots, send them for mass spectrophotometry and protein identification, and then we did the bioinformatic analysis. This is, for example, when we do this isoelectric focusing, what we do is we use different steps, right? So we use different programs. These are the gels of my student, which she standardized, and it took her 8 months to standardize this program. Okay, and then she did 2-dimensional gel electrophoresis of proteins, and this is the photograph of her computer screen wherein she is analyzing all these differentially expressed proteins in Gujarat maize and Gurez local in response to low temperature. So what she did, de-staining of the protein spots with the gel, then reduction and alkylation. These are different steps. Then trypsin digestion, extraction from the gel, and then we sent it for the Malditov. We outsourced it at CDRI Lucknow. And okay, this is the different steps, flow chart of the workflow. Okay, and the different softwares which she used to analyze these spots, SOPMA, Fire2, ModWeb, 3DSum, etc. These are, for example, Gurez local at 0 degrees, Gurez local after 2 hours, 6 hours, 10 hours, and 12 hours. Similarly, Gujarat maize, 0 hours, 2 hours, 6 hours, cold stress, 10 hours, and 12 hours. So we compared it, right? We compared which proteins are getting overexpressed, which proteins are getting down expressed. So upregulation, downregulation. So we'll see what is missing in these or what is extra in these. That will be critical for cold tolerance, or those proteins are important for low temperature stress. So this is the representative 2D gel from each of the genotype, which depicts how proteins were selected. Okay. So Salika found 12 proteins which were downregulated and 28 proteins which were upregulated. This is the mascot research results. Okay, these are the screen graphs. Okay, and this is the table wherein we have this on the base of Malditov. These are the differentially expressed proteins. This is unpublished data. We have communicated it. We have communicated it for in different journals. So let us see. Let us hope that our work gets published. Okay, briefly. So this is differentially expressed proteins of Ziamase identified by Malditov MS. So these are highlighted. For example, threonine dehydratase biosynthetic chloroplastic protein. We found that it is getting overexpressed in Gurez local, and it does not get overexpressed in Gujarat maize. Similarly, there was another unique protein, GINS complex protein. We reported it from Ziamase that in Gurez local that it gets overexpressed. Similarly, F10K1. And very interestingly, we found nodulin-like protein, which has not been reported so far. We are publishing it. that this protein gets overexpressed in response to the low temperature. It has not been reported so far. Similarly, we have Gurey's local genotype. For example, one protein got downregulated. Spot ID is this. Okay, we get spots in the protein, and we saw that this protein is Ziamase GINS complex protein. So this is SOPMA analysis file. Okay, I'm taking much time.
Yeah. I think you have 2-3 more minutes if you want to take.
Okay. Thank you.
Maybe 5 minutes per question.
So, okay. So these are different differentially expressed proteins we identified. We try to identify. So that is I will conclude it. Maybe they will not try to make out what is this. So we have identified couple of proteins not reported so far. We are going to further characterize them, further work on them, and publish the paper. Out of total 29 spots identified, 10 proteins were detected in low temperature sensitive Gujarat maize, and 19 proteins in low temperature tolerant Gurey's local. Right. So we found 10 in Gujarat maize, 19 in Gurey's local. In Gujarat maize, 5 proteins showed upregulation and rest 5 spots got downregulated. Means they might be, they might be repressed, or they might be degraded, or they might be repressors, whatever. So similarly, Gurey's local, we found 17 proteins exhibited upregulation. Usme humne ye dekha, only 2 were downregulated in response to the low temperature. New report of 4 proteins of having exceptional role in low temperature stress tolerance have been identified in the Gurey's local. And for these 4 identified proteins, there has been no report so far in the literature, which we did. So we are going to further characterize them, and we are further going to take them forward. So thank you so much for your patient listening. Yeah.
Thank you, Dr. John. Yeah, it was interesting to know how technology is actually contributing for a sustainable development, I would say, like feeding lots of mouth at the same time and taking care, that there's a balance and harmony with the environment. So that was something really very, very interesting. And the session is open for questions now. If students have specific questions related to talk, or maybe related to the career which Dr. John has right now, maybe if you have questions how to get into this field and what kind of profile you need to build up. So the room is open for any kind of questions or discussions you have. We have one question in chat window. Meantime, students can type their questions in chat, or they can raise their hands. One question, Rifat, is from Keithana.
Yes.
The question is, can vertical farming help cover the shortage of arable land, or is it viable only for small scale decorative plants?
I mean, why not? I mean, vertical farming can also help us. What does, what the idea is that, since I said that arable land means that land which can be brought under the agricultural use. So we have a land which is arable, which we use for agriculture purpose. So the idea is to use non-arable land, that land which normally using the spaces for agricultural produce, which conventionally were not being used for agricultural produce. So yes, vertical farming can also be used. We can use every space, every nook and corner, for agricultural produce. For example, when we talk about the bioenergy, we talk about growing algae in the oceans for bioenergy. So we can use any space, that is by using modern technologies. We can use any space, anywhere, grow plants, not only for food but for oxygen. Yeah,
thank you. And I'm just curious, are there any kind of fellowships or internships for, like, most of the students in the group are undergraduates or maybe from school, like 11th, 12th standard. So are there any say funding options, or maybe internships options, not even funded, which students can opt for and try to make a profile for in this field?
Bindya, I must tell you that this, as I said, that this global hunger and agricultural production, these come under the United Nations program, and this is the priority of our present Indian government also. from time to time we get calls for fellowship, or there are schemes, but they are not, I mean, throughout the year open. In between, there come calls, apply if you have an idea for agricultural produce, or if you have an idea for, we talk about robotics in agriculture. You have a large agricultural field, and then see, you have to use the fertilizer. Now you cannot go, that is labor intensive. Now, how to use robotics to spray, fertilizers, nanotechnology, it's not that, how to use nanotechnology for improving the agricultural production. So from time to time, there come fellowship programs. There are schemes for scientists, for students, wherein they are asked to submit their ideas, submit their proposals, submit their thoughts, so that screening committee evaluates and then, grants fellowship or grants the projects. As such, there is no separate. Yes, we have, for example, Ministry of Environment and Forests. They grant projects. Yeah, you can, students, if they are interested, they can contact Ministry of Forest and Environment Resources is there. So there are many ministries there wherein students can, ask for the information. But as such, I don't find any particular fellowship for UGs related to agriculture. With agriculture, we have an institution, ICAR, which deal with agricultural programs. Yeah.
Another question is for Antarpreet. She asks, what is scope after MSc Botany other than going into research field?
Okay. So see, we have also a PG student. Let me be very realistic. What students do here is, I have a couple of options which I tell students, being because I was academic counselor. Number one is to pursue very good hands-on research experience outside the India, right? They call for PhD programs, they give fellowship, right? And get a very good hands-on experience and come back to India and apply for very good position. That is one. Another is, I have seen students preparing for civil services. Once they have an administrative position. We have many students from our department who became administrative officers, and then they utilize this agricultural knowledge or this botany knowledge in their relevant field when they are in administration, and they are more environmentally sensitive. And number three, they can have their entrepreneurs. Bindia, I have seen 2 or 3 ladies, I don't remember the name. You know what they did? They grew the lavender gardens, lavender trees, and they are making huge money. They are entrepreneurs for this fragrance. They have a huge market. Floriculture, it is booming. People grow those exotic, for example, you want a plant which grows in Europe but does not grow in your place. Now what you do is you create such soil and polythene, and they polyhouses they grow exotic vegetables which are good for health, organic, and that's a wonderful career also. So opportunities are immense. There is no dearth of opportunities. It only is resilience of a person to pursue aim. So botany, I think, has an immense opportunities as an entrepreneur, as an administrator, as a researcher, as a teacher, or as a social activist also.
Yeah, that's wonderfully put. Thanks so much, Rifat. And
thank you.
I think there are no more questions. It was a very interesting talk.
Thank you.
I would request everyone present. Most of them have left. There were around 30 during the talk.
Yeah, it's okay. We had 2 years of COVID, and then we used to teach all this, Zoom.
I request everyone present to kindly switch on their camera so that I can have a virtual group image, like a screenshot.
If they are around. Okay.
Hi, yeah.
Hello, everyone. Nice to see everyone.
And I can understand the anxiety, like when you're presenting a talk, you have to put, is everybody is there, or you are listening?
I'm mute. in between the classes, I used to tell, I'm audible, am I audible? Because I thought everybody is sleeping. It's me who's talking. There were memes on the internet about this. Some Japanese teacher who kept on giving the lecture for 1 hour and he was on mute.
Sometimes it happened that I got disconnected and I didn't know that I wasn't in the meeting, and I just kept on giving the entire lecture. So others can also kindly switch on their cameras. I need at least 2-4 people more to switch on their cameras.
To complete that screen.
Yes.
They are coming.
Please.
All the best, everyone.
Okay, I'll take a screenshot. 1, 2, and smile.
Thank you.
Great. Thank you so
Thanks,
much.
Bindya. Thanks, Bindya. Thanks, everyone. Thanks for patient listening. Thank you all.
Thank you.
Bye.